So in today's lesson, we are going to look at vaccines. Before we start talking about vaccines, we're just going to have a quick recap in what we did in the previous lesson because it ties in. So in the previous lesson, we looked at the immune system. We saw the immune system was responsible for fighting infection. So our body has lots of defenses to stop pathogens getting inside. So we're covered in skin. We've got mucus in our nose and down the back of our throats. We've got acid in our stomach. It's got lots of defenses to stop pathogens like viruses and bacteria from getting inside and making us sick. But inevitably, some pathogens are going to get inside our bodies. And when they do, it's the immune system that will try and fight that infection. Now, there are lots of different aspects of the immune system, but the one that we focus on is the white blood cells. It's not the only part of the immune system, but it's the part of the immune system that we focus on, the white blood cells. So I'll just write it here. Part of the immune. They're part of the immune system. And we learned in the last lesson that there are two types of white blood cells with very different jobs. So we had phagocytes as one type of blood cell and lymphocytes as another type of white blood cell. So you can say overall white blood cells fight infection, but then you want to be more specific. There are two different types of white blood cells. They're both involved with fighting infection, but they do so in different ways. So phagocytes, we say that they're non-specific and they will fight any pathogens that get inside our body. And what they will do is they will surround and engulf or destroy pathogens. Okay? The lymphocytes do something quite different, okay? They're still trying to fight pathogens, but they do it in a different way. And what they do is they produce antibodies. Okay, they produce antibodies. And what antibodies do, and again, I did go through this in detail in lesson 7, I think it was. If you haven't watched that, please do, okay? So they produce antibodies, and antibodies will recognize antigens on the surface of pathogens. Now, I know I'm using lots of key words here. I've got antibody, antigens, pathogens, lymphocytes, phagocytes. There's a lot of vocabulary in this topic, and it's really important you know the difference between the key words. I'm going to draw a diagram in a second to help you out with this, okay? But lymphocytes are the white blood cells. They produce antibodies. Antibodies recognize antigens on the surface of pathogens, and pathogens are destroyed. Okay, so I'm going to draw a little diagram here of a lymphocyte. Okay, so a lymphocyte is our white blood cell. So I'm going to put a little smiley face on my lymphocyte because they're there to help us out. And I'm going to draw a pathogen, and I'm going to put a little unhappy face on my pathogen because they cause disease and they are not doing us any favors, okay? And it says that there are antigens on the surface of pathogens. So that just means on the outside of the pathogen, you have got these little molecules called antigens, and they come in all sorts of different shapes and sizes. I'm just drawing it like a Y shape, but these are called antigens. And you find them on the outside of pathogens. And what the lymphocyte will do is they will recognize these antigens and they will start to produce antibodies. And they will produce antibodies in a complementary shape. What I mean by that is here I've drawn the antigens as a Y shape. And then on the lymphocyte, I've drawn an antibody in an arrow shape. And what this will do is the arrow shape is complementary to the Y shape. The arrow shape will fit into the Y shape just like a lock would fit into a key. And that's really important. The shapes have to be complementary. They have to match and fit into one another. So I've got my antibodies and I've got my antigens. So this is my lymphocyte, my white blood cell. It produces antibodies that are complementary to the antigens on the pathogen that has gotten inside your body. And because they're complementary, what will happen is these antibodies will bind onto the antigens. And when the antibodies bind onto the antigens, then the pathogen is going to get destroyed. Okay, and it won't be able to make you sick. So by producing these specific antibodies, they will bind to the antigens on the pathogen and the pathogen will be destroyed. Okay? The only problem with this response is quite slow. So when you get a pathogen inside your body, especially if it's the first time that pathogen has ever entered your body, it's going to take a little while for your lymphocytes to make the right antibody that will perfectly fit with the antigen on your pathogen. So this will take a little bit of time. So I'm going to put in brackets here that this whole process is a slow response, okay? It takes a little bit of time for your lymphocytes to make the right antibodies that are going to lead to the destruction of your pathogens. And while you're waiting for your lymphocytes to produce the right antibodies, you're feeling sick. Okay, you're feeling quite sick. You're being affected by the disease. So if it was a bacteria that you had in your body, that bacteria would be producing lots and lots of toxins and making you feel sick. If it was a virus you had in your body, that virus would be getting inside your cells and destroying your cells. And these things are going to make you feel sick. So while you're waiting for your lymphocytes to start destroying those pathogens, you are feeling quite sick. And it can take quite a few days before your body learns how to respond and learns how to fight and kill the pathogens. And then you've got some cases where your body just doesn't learn to fight it. You get hit with such a big load of the pathogens. You get infected by quite a lot of the virus or a really difficult strain of the virus right at once, and your body doesn't learn to fight it quick enough, which is obviously what's happening at the moment in some cases with the COVID-19 virus. That's why it's causing deaths. People aren't fighting enough quick enough, and they're dying before their body has learned how to fight this virus. Okay, but while the initial response to the pathogen is slow, okay, so I've said this is a slow response. The good thing about it is that even after the pathogen is destroyed, so your body's made all of these antibodies, and the pathogen is destroyed, you start to feel better. Okay, as soon as you start to feel better, and those pathogens have been destroyed, you don't need the antibodies anymore. They're there to destroy the pathogens. When the pathogens are destroyed, you don't need them anymore. But luckily, those antibodies don't just go away. They will remain in your system. So all those antibodies that you produce, you won't have as many of them because you won't need them, but they will remain in your system. And because they remain in your system, okay, because these antibodies don't completely go away, if you get infected with that same pathogen again, because you already have the antibodies in your system, you will fight that pathogen a lot quicker the second time. Okay, and in some cases, so for example, something like the chickenpox, you may know that the chickenpox is a disease that you typically get once and then you don't get it again, okay? That's not because you never come in contact with the chickenpox virus again. That's because if you've had it once, those antibodies that your body produced to fight the chickenpox virus, they remain in your system. And the second time you get infected with that same chickenpox virus, your body fights it so quickly that you don't even realize you were infected in the first place. You don't get the full disease again, even if that virus gets into your system. So having those antibodies in our system will prevent us from getting sick if you get infected again. And we say that the antibodies remain in our system because we've got, and again, I do explain this more in the other videos if you haven't watched it, okay? The reason that we can produce these antibodies so much quicker the second time is because you've got what we call memory lymphocytes. So I'm going to draw the other way. I'm going to draw my lymphocytes again, and I'm going to call it a memory lymphocyte. And by that, I mean, after you've been infected once, your lymphocyte, which is your blood cell, remembers how to fight pathogens. It will stay inside your blood and they will fight a lot quicker the second time. So I'm going to say you get a faster response because your body has fought the disease once before. It will fight it a lot quicker the second time. Okay, so I did say the title for the lesson was vaccines, and I haven't actually mentioned yet what a vaccine is. Okay, so underneath we are going to write down what a vaccine is, and then I'm going to explain its purpose. So vaccine, which is spelled with 2 C's. So a vaccine is an inactivated, which is a weakened form of a pathogen. Okay, so I'm going to talk about the idea of vaccines. Okay, so we've seen that if you get infected with a pathogen the second time, your body can respond a lot quicker, and you can respond so quickly that you don't even feel sick. The idea behind a vaccine is teaching your body how to fight certain pathogens in a safe way without you getting sick at all. So if you look at the definition for a vaccine, you will see that it's an inactivated or a weakened form of the pathogen. So what they would do, for example, if you had a chickenpox vaccine, they take the chickenpox virus, okay, so the virus that causes the chickenpox, and they would do something to that virus. They would inactivate it or weaken it in some way. And once they've inactivated it, they would then inject that virus into your bloodstream. And the point of that is so that your body can learn how to fight that virus. So remember, you've inactivated it or you've weakened it. So it's not going to be strong enough to be able to infect you and to make you sick. But that inactivated, weakened form will still have the antigens, okay? It will still have those specific markers on the outside of the virus. And what your immune system can do is your immune system will learn to recognize those antigens, and your immune system will learn how to make the right antibodies, okay? And remember, because it's in an activated form or a weakened form, it's not going to make you sick, but it teaches your immune system how to produce the right antibodies. Then later on, if you get infected by the virus for real, your body will already know how to fight it and will be able to make antibodies so quickly that you're not going to feel sick. So underneath we're going to write, so we've written that it's in an activated form of a pathogen. I'm going to write that it causes our white blood cells, and I'm going to be more specific, and I'm going to put lymphocytes in brackets because they're specifically the type of white blood cell that we are targeting here. It causes the white blood cells to produce, all right, complementary antibodies. So they're not just going to start producing lots of random antibodies. Yes, you have inactivated your pathogen, but it still has the same antigens, and your lymphocytes will start to produce the right complementary antibodies. To produce the complementary antibodies, okay? You're basically teaching your body how to make those antibodies, and what that will do, then I'm just going to write underneath, if you're infected with the real virus, with the real pathogen, so you get properly infected with the full form of the pathogen, it's activated and it's strong. If you're infected with the real pathogen, your immune system, and specifically your white blood cells and specifically your lymphocytes, can fight the disease quickly, which will stop you getting sick. Okay? So basically what a vaccine will do is it will train your immune system to produce the right antibodies. Those antibodies, some of them will remain in your bloodstream, and you get infected with the real pathogen later on, and you will be able to respond so quickly that you won't feel sick. So for example, measles used to be a really big problem. Now, measles is quite rare in this country, okay, because people have got the measles vaccine. They get it when they're children, you get the measles vaccine when you're a kid. Your body learns how to fight measles. Because everyone in the population is able to fight the measles virus, no one becomes sick with measles, and then nobody spreads it on. Okay, so I'm going to turn the page. We've got a few other little things to jot down, and we're going to look at some questions on vaccines. So I'm just going to write here, vaccinations lead to immunity. You might be familiar with that word, immunity, okay? Immunity means that you're not going to be affected by the disease. You're immune to the disease, you're not going to be infected, you're not going to get ill. And we say vaccinations lead to immunity because if everyone in a population has the vaccine and no one is getting sick from the virus, then the virus isn't going to be able to grow, it isn't going to be able to spread. Okay, so again, if you think of the flu virus, if you think of how the flu virus spreads, okay, the flu virus is airborne. If I have the flu virus and I've got the flu, one of the symptoms of the flu is that I'm going to be coughing and sneezing. And as I'm coughing and sneezing, each time I cough and sneeze, I'm spreading that virus into the air. So by not being able to fight the flu, I'm getting sick myself, but it's also giving me symptoms that lead me to spread this virus to other people. On the other hand, if I was able to fight the flu, if I had my vaccine and I was able to fight the flu, then my body would kill that virus so quickly that I wouldn't get those symptoms. I wouldn't be coughing, I wouldn't be sneezing. My body would destroy the virus and I wouldn't be spreading it on to anyone else. So if you've got a whole population where everyone can fight these pathogens as soon as they enter their body and they're not spreading them, then you end up with immunity where the diseases just die out amongst a population and they're no longer a problem anymore. So vaccines lead to immunity. I'm sorry. All right, that you're no longer affected by the pathogen. It's not that the pathogen cannot get inside your body, it's just that when it does, it's not going to make you sick. You're not going to be affected, you're not going to get sick. Okay, and they can lead to immunity. That's pretty much all you need to know about vaccines. You just need to know what they are, okay, an inactivated form of the pathogen, and you need to know the purpose of vaccines, okay? It's to teach your body how to make the right antibodies so that you can fight the disease really, really quickly if you get infected, okay? And you've all had vaccines already. They come into school and they give you different vaccines, okay? They're typically given by injection, but they can be given also by oral tablets, so a tablet that you would take as well. Actually, just one more thing that we're just going to jot down really quickly and then we'll do some questions. So I'm going to draw a graph, and we drew something like this in the last video, something quite similar, okay? On the y-axis, I'm going to put antibody concentration. So how much antibody you've got. Antibody concentration in the blood, so how much antibody you have got in your blood. And then down on the x-axis, I'm going to put time, but I'm going to do it in weeks this time, not just days. So I'm going to go 1 week, 2, 3, 4, 5, 6, I'll make it a little bit longer, 7, 8. And this is time, and it's in weeks, okay? And we are going to have here, time is T0, we are going to have your vaccine. So this is when the person is getting their vaccine. So they are being injected with a small amount of the weakened pathogen. And when that weakened pathogen enters your body, what you're going to see is that slowly your body will start to produce antibodies against that pathogen, and the antibody concentration of your blood will rise slowly, 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 and then it will level out. And then as you fight off the vaccine, it will start to drop back down, but not completely back down, but it will start to drop back down, okay? Then, say a few weeks pass and everything is fine. And then, okay, we're going to call you week 5. At week 5, you get infected. So you get infected for real by the pathogen. And what you're going to notice here is you get a... Okay, you're going to see two massive differences in the response. Okay, when you got infected with the vaccine, this curve here was quite gradual. Okay, it took your body time initially to increase the amount of antibodies in your blood. So that was a slow rise, and it didn't rise particularly high. When you got infected for real, you get a really, really sharp increase in the amount of antibodies in your blood. Okay, your body didn't have to learn how to produce the right antibody. It already knew how to produce the right antibody. So instead of having to wait kind of 2 weeks before you had lots of antibodies in your blood, you were able to produce them straight away. You get this really sharp line, this really sharp increase. Okay, so we have a gradual increase. And here I've got a sharp increase, and I can tell by the steepness of the line. Okay, this line here is much steeper than this line here, which tells me I have a much sharper increase than what I had over there. The other big difference that you'll notice is that when you had the vaccine, the levels of the antibody in your blood was not that high, but when you got infected for real, you had a much higher level of the antibody in your bloodstream. Okay, and it'll be up there, and then when you fight off your pathogen, then those antibody levels will start to drop back down again. Okay, but you had a higher concentration, a higher total concentration when you got infected for real than when you got when you got the actual vaccine. Okay, we're going to look at a few questions now, a few exam questions, because they've asked lots and lots of questions about vaccines and vaccinations. It's a really, really common exam question, but luckily they ask the same thing every single time. There's only so many things that they can ask you about vaccines, and the questions tend to be very, very similar. So if we just do 2 or 3 questions together, that'll pretty much cover you for all of the exam questions that they're going to ask you about vaccines. They do ask some 3, 4, even 5 mark questions about them, but they are all pretty much the same. So I'm going to put questions on the screen. I want you to answer them. So pause, answer, and then press play when you are ready to have a go yourself. So this is, oh, that's very bright. Boom. Okay, that you should be able to see. So this is question number 1. The first one, you're just matching the disease to the way the disease is spread. It's 3 marks, so 1 mark for each you get right. And then at the end, something that we did in a previous lesson, 2 ways that the human body protects itself against the entry. So specifically, how you stop the pathogens entering your body, not how you destroy them when they're in your body, but how do you stop them entering your body. Press pause, answer the questions, and then press play. Okay, so there's your answers. Cholera, it's a bacteria. It's spread in dirty water. The cold, like the flu, is spread airborne when people cough and sneeze. Malaria, spread by mosquitoes, in this case animals that draw blood. You're asked to give 2 ways that the body protects against the entry of pathogens. So I've written skin acts as a barrier. Mucus traps pathogens. You could also write nose hairs trap pathogens. You could write that your platelets clot the blood and form scabs to stop pathogens entering through your skin. You could write stomach acid, so hydrochloric acid in your stomach. The antimicrobial chemicals that they're referencing here would be lysozyme, so you wouldn't get a mark for talking about lysozyme, but any of the other defenses that stop them entering, you'll get a mark for. Okay, so question 1, D, E, and F. So I appreciate that this might be a little bit hard for you to read, but just do your best and try and be as accurate as you can. They want to know what day the second vaccination was given, the highest concentration produced by the first antibody, and then have a go with F. I appreciate you probably can't see the figures well enough to do the first 2 questions, but have a rough guess and at least do F for me. So press pause, do the questions, and then press play again. So here are our answers. They want to know the day the second vaccination was given. So they tell you the first vaccination was given here at day 0. The second vaccination, okay, I can tell because I see an increase in the amount of antibodies produced. And this second increase starts on day 28. So the second vaccine was given on day 28. So I can see right here that it's decreasing, decreasing, decreasing. And at day 28 is when it starts to increase. So I appreciate you might have been able to read that from your screens. Highest concentration of antibodies produced by the first vaccine. So this was my first vaccine. I see an increase in antibodies, and the highest it gets to after the first vaccine is 2.5. You can see then that it decreases, and after the second vaccine gives you a higher total concentration of antibodies in your blood, which is why that second vaccine was necessary in the first place. Then the effect that the vaccine is going to have, you're going to get a decrease in the number of children with the measles virus, and the reason that as everyone's vaccinated, you're less likely to come into contact with someone that has the disease. Therefore, you're going to be less likely to catch it, and the numbers are going to decrease. Question number 2. Again, we're still talking about measles. I think, have a read through the questions. I think they should be self-explanatory. I'm going to press pause, press play when you're ready to see the answers. Okay, so the answer, the first question, they want to know why the actual number of cases of measles might be higher than in the table above. A simple reason is just not everyone goes to the doctor. So it's like what we're having at the moment with the COVID-19. And, they're saying that the numbers that they're reporting of infections and of deaths in the country are not entirely accurate, because some people haven't gone to the doctors, they haven't been diagnosed, they could have, they could be infected at home and not have been tested, they could have, died at home or in care homes and not have been tested. The same with measles. Some people who had the measles didn't go to the doctors, their case was never recorded, and so the numbers are likely to be lower. So these are only the numbers of confirmed cases, but those numbers are likely to be higher. Calculating percentage increase or decrease. I think we did percentage increase last time, but percentage decrease is going to be exactly the same. They're asking you to calculate it between 2012 and 2015. So the first thing I need to do is calculate the change between 2012 and 2015. So in 2012, you've got 2,030 people with the measles. In 2015, you've only got 91. So that change in the number of people with measles over those 3 years is 1,939 people. So I take my change and I put it over the original number to calculate the percentage decrease. So it's going to be 1939 over the original number of 230, and then you multiply it by 100 and you get 95.5%, or you can round that up to 96%. That's percentage decrease, so give yourself a full 2 marks if you got either of those 2 answers, even if you haven't shown me they're working. And then the last bit is similar to what we've seen before. If you vaccinate, why will that reduce the spread of the measles? If you vaccinate, most people are going to become immune. So even if they come in contact to the virus, they're not going to become ill. And that means that the people that aren't immune, that haven't been vaccinated, they are far less likely to come in contact with the disease if the rest of the population is immune and will not have the disease. So anything along that lines will get you your marks. Okay, we're going to just do one more. We're going to do a 6-mark question together. So this is a full 6 marks. I'm going to show you the whole thing. So the graph below shows the concentration of measles antibodies in the blood of a boy. Explain the difference between antibody production after the vaccine injection and after exposure to the measles virus. Okay, so showing you a graph like what we drew in our books. This is when he's actually getting the vaccine, and you can see that the amount of antibodies in his blood increases after he gets the vaccine. Then a couple of weeks later, he gets exposed to the measles, the actual measles virus for real, and then you can see how the antibody levels in his blood change. And what they want you to do is to explain the difference between antibody production between the vaccine and the actual exposure to the virus, and give data from the graph below. So just explain the differences, just like we talked about below, between the steep increase or the sharp increase and the gradual increase in terms of the actual number of the antibodies produced in each case, and then try and give me a reason why the responses or the amount of antibodies in the blood is so different. Pause, I'm going to write out an answer and then press play when you want to look at it. Okay, so I've written out an answer. The first part of the question, they just want you to comment on the differences, and then they want you to be able to explain the differences. And it does say that you should include data from the graph in your answer. And I appreciate that that might be difficult to do reading this off a screen, but there are a few kind of general observations you can make. So the first difference I'm going to say is that the antibody concentration is much higher after he was exposed to the virus compared to after he was exposed to the vaccine. So this was the response, okay, when he got exposed to the vaccine. But this was the response after he got exposed to the real virus. So just saying antibody concentration, which is what's on your y-axis, was much higher after he got exposed to the virus than the vaccine. Okay, and it says to include data from the graph above. So I've said it's 7.2 compared to 0.8. And they haven't given you proper units on the y-axis. They just said in arbitrary units. Okay, so you don't have to write down grams or milligrams or anything like that. So I'm just going to write 7.2 was the highest point for the virus. Compare that to 0.8 was the highest antibody concentration for the vaccine. The second thing I commented on was I said that the antibody production was much quicker after he was exposed to the virus than the vaccine. And the way I know this was because the virus curve is really, really steep, whereas the vaccine curve was a lot more gradual. So I just wrote underneath steeper curve. So I've given two differences between the two responses. And in each case, I've referred back to the data to back up my answer. The other thing you could potentially say in terms of your differences is you can say that for the virus, that the antibody concentration remained higher for longer. So you see with the vaccine, it comes down again quite quickly, but with the virus, that antibody concentration stays high for longer. So you could also mention that in your answer. So they're the two main differences from the graph, but now you need to be able to explain those differences. So why did I get a higher antibody concentration, and why were those antibodies produced even quicker when he got exposed to the virus compared to the vaccine? Okay. And it's all because when he was exposed to the virus, it was his body's second time encountering that particular pathogen. And because it was the second time, his memory lymphocytes were ready and they recognized the antigen on the pathogen and they were able to produce the complementary antibody quickly. I've used a lot of our keywords there in those last two sentences. You don't need to use all of them. You would be okay if you just said the white blood cells could make antibodies. But do try and use as many of those keywords as possible. Okay, so because it was the second time encountering this pathogen, his white blood cells were able to respond much quicker. His white blood cells were able to produce antibodies much, much quicker. And I've just been more specific by saying it was specifically memory lymphocytes. They're the ones that remember that they recognize the antigen on the pathogen. And then I've said they produced complementary antibodies quickly. So I've used a lot of the keywords. Try and use them where you can. But if you've just written something more general, that the white blood cells were able to produce the antibodies, then that's fine. Okay, just make some sort of reference to the fact that he's been exposed to this pathogen before, which is why he was able to fight it so much quicker the second time. Okay, so we'll leave it at there for vaccines. The next lesson we're going to look at antibiotics. So you're probably familiar with antibiotics, but we're just going to look at a little bit more detail what type of pathogens they treat and exactly how they work. And then we're going to look at drug development in general and clinical trials and how you get a drug from the lab and from scientists working on the lab and being actually administered in a clinical setting. So we're going to do that in the next lesson, as well as having a look at a couple more exam questions.